Hello everybody, and welcome to the lecture on the cell membrane as well as passive and active transport. So these two processes occur across the membrane, so it makes sense to put them together as one lecture. This first slide is going to go over some stuff that we've already talked about, but it's important to make sure that it's ingrained in your brain before we go into any more detail. So, number one, cell membrane. Whenever you have a cell, the outer boundary is defined by the cell membrane. Right? In terms of its structure and how it's built, this goes back a couple of lectures. It's 2 molecules thick, and if you were to magnify a little section of it like this, you would notice that it looks something like this. Okay, so let's just real quick set up the environment of the cell. Within the cell, you've got a lot of fluid called cytosol and a whole lot of organelles doing their jobs, and all of that together is called the cytoplasm. Outside of the cell, it's mostly a watery environment, and it is referred to as the extracellular matrix. And so you've got the cytoplasm inside with all of the organelles doing their job, the extracellular matrix outside the cell, and separating those two is this little double layer of molecules. The name of those molecules is phospholipids, and it's 2 layers of them. If you remember back, the heads, which is the part that I'm coloring in black here, those are water-loving, they're hydrophilic, and so they face out toward the extracellular matrix and inward towards the cytoplasm with the double layer structure that you see. That's because those hydrophilic heads want to touch water. The hydrophobic tails face inward so they don't have to touch water. So that is the setup of the cell membrane, and this is kind of the environment that we are dealing with this chapter, at least at the start here. Another important thing to note, which you see up here, plasma membrane has the exact same meaning as cell membrane. So those two terms are going to be interchangeable. So what is the purpose of the cell membrane? First and foremost, it defines the boundary of the cell. It serves as a protective barrier for the structures within the cell. However, there's a whole lot more that's going on here. If you remember back to the previous lecture, cells were small because it gave them more surface area. It gave them more surface area of cell membrane. And that's important because there are a lot of things happening within the membrane. Right? The cell membrane performs a variety of functions for the cell. And I'm going to go ahead and click a couple of slides forward. Here we go. This picture, I think, shows some of the functions of the cell membrane very, very well. So this picture, it's showing 2 cells. So hypothetically speaking, this picture is looking at right here. We're looking at 2 cells neighboring each other, right? And you've got the phospholipids making up the structure of the membrane, but you've got all of these things embedded within it also. So what are those things? Those are proteins, and they're doing various functions for the cell. So let's just take a look through at some of these proteins and what they're doing. Let's go from right to left. This is called a glycoprotein. It has a little sugar chain right here. This sugar chain is unique to this cell. It's almost like a fingerprint in that sense. It's going to be recognized by another cell. This other cell is going to recognize the glycoprotein, and so it will realize what this cell is and respond accordingly. So cell-to-cell recognition can occur because of proteins within the cell membrane. Junctions, joining cells together to make tissues, those are proteins that occur within the cell membrane, like you see here. Regulating transport of molecules in and out of the cell, you can see that occurring here. Receiving signaling molecules like hormones, and then responding accordingly, that occurs with proteins that are embedded in the membrane. And there's a couple more here that we're not going to talk about, but you can see there's a lot going on in here. There's a lot going on. And cells being small gives more surface area for all of this stuff to happen. So the cell membrane performs a variety of functions. Here's a couple of them that we talked about with that picture that we were looking at. In terms of the structure of the membrane, the phrase that gets used is fluid mosaic model. And I can explain it to you, but I think this one is best observed in a video, which won't be very good quality if I try and play it and record it on my phone here. So what I've done is I've linked the video on Canvas for you to watch. It's 2 minutes. It summarizes the fluid mosaic model very well. So I'm going to still explain it here, but I recommend that you watch it because it will help you in understanding. So a mosaic, in this case, refers to a variety of things. In this case, a variety of molecules embedded within the cell's plasma membrane. So if you look at this slide again, we've got a whole mosaic of different proteins embedded within there. Plus we've got cholesterol, sometimes there's sugars in there too. All of these molecules within that membrane constitute a nice mosaic, a variety of different things. Cholesterol's function, by the way, which you can see it right here, its function within the cell membrane is to help make it fluid at different temperatures, and so that's what its job is going to be. So the cell membrane is a mosaic of different molecules. The fluid part of the fluid mosaic model means that the proteins don't just sit cemented in one spot. And the video that you've hopefully watched shows this very well. It shows the proteins moving around in the membrane pretty fluidly. And so the fluid mosaic model explains pretty well the nature of proteins and other things that are embedded within the cell membrane. All right. Shifting gears, now that we've got a good understanding of the nature of the cell membrane, now we want to talk about the movement of molecules across it. So the cell membrane regulates the movement of molecules into and out of the cell, and only certain molecules are allowed to cross. Other molecules maybe can't cross because of size, or the cell actively works to keep them out. Either way, the cell membrane allows certain molecules to pass, but doesn't let others pass. That is called selective permeability. So be aware of that term, because we're going to revisit that when we talk about something called osmosis, which we'll get to in a little bit. But now we got to talk about diffusion. And this is a very important concept, and it can be a little tricky sometimes to understand at first. So, I'm going to draw a cell up here, and I'm going to use the red marker to represent a particular ion. We're going to say it's potassium. Now, I don't know how well you're going to be able to see this on the phone. I hope you can see it well. But I've got a bunch of red dots within the cell. So this black line is the cell membrane. Right? I've got a bunch of red dots within the cell. I've got a few red dots around the cell. The red dots are going to represent a potassium ion. These exist within your cells and in the fluid surrounding them. So, we've got a whole bunch of potassium ions within the cell. We've got a few of them outside the cell. What's going to happen, and let's assume that the cell allows the potassium ions to cross. Remember, it allows certain things to cross. We're going to say that it allows potassium ions to move in and out of the cell. So if you were to have this cell, and this was your starting point in terms of potassium ion distribution, if you were to just let this thing be, let time pass, what's going to happen is that there's going to be an overall movement, a net movement of potassium ions out of the cell. Because there's a lot of them in here, there's not a lot of them outside the cell. And since they can cross that barrier, they're going to move from a higher concentration inside the cell toward the lower concentration outside the cell. And you're going to have that net movement outward until we've got an equal number of potassium ions inside and outside. And once that point gets reached, it's called equilibrium, and then you're going to have equal movements of molecules going in and out. There's not going to be any high to low movement, because there is no high to low anymore. We're all equal. If the potassium ion example doesn't help, hopefully this picture will. The gray line here, this dotted gray line, that is a cell membrane, and these green circles represent a molecule. Clearly, there's more of them on the left side of the membrane than the right. So if you were to let this be over time, you would have a trend of movement towards the lower concentration until equilibrium gets reached, and here we have it with 8 molecules on each side. The molecules will still move back and forth, but now it's going to be equal. Before, it was not equal, and so they had a net movement towards the right. So, time to apply some terms to all of this. The movement of molecules across the cell membrane from high to low concentration. High to low concentration. This is called diffusion, aka passive transport. These two terms generally have the same meaning. So, diffusion or passive transport is the movement of molecules along their concentration gradient from high to low. The key with this is that it is passive, which means you don't need energy to make it happen. If you were to just let it be with no energy input over time, it's just going to trend toward equilibrium until it gets there. The only way that you prevent molecules from moving high to low and going toward equilibrium, the only way you stop it is by putting energy in to stop it. So over time, this is just going to occur. Passive transport, movement of molecules across the membrane from high to low concentration happens over time without any energy input, and it occurs until we get to equilibrium. And then, like I said, it's equal movement both ways. Okay. All right, I want to go back to this picture. Certain molecules can clearly cross the membrane, but they often don't want to try and cross through this maze of phospholipids because it's just kind of messy and not very efficient. And so instead of trying to squeeze through here, what your cells will do is they will have a channel protein, like a tube, through which molecules can easily pass. This picture is showing how that works with water molecules. So water molecules, they don't want to try and squeeze their way through this hydrophobic interior of the membrane. And so you've got a channel protein that clears space for them to come through. Water so abundantly moves across the cell membrane that they have specific channel proteins that are made just for them. They're called aquaporins, literally means water pore. And so aquaporins allow water to move across the membrane through a channel protein so they can move much more efficiently. In fact, it causes water to move hundreds of times more quickly than it could without them. If you've got a channel protein like that, providing a pathway, it's still diffusion, but it's called facilitated diffusion. But the key there is that it still doesn't use any energy. Diffusion is always going to be passive, no energy required. Speaking of the movement of water, there's a term specifically for the movement of water across the membrane. That word is osmosis, and this is a very important concept when it comes to living things. The picture in the PowerPoint there is a pickle. I like pickles. Pickles are made from cucumbers, and I've never made a pickle, but my understanding of the process is that it involves placing them in a certain type of solution that includes vinegar and salt. So let's go ahead and show how that would work. Here's our cucumber. I don't have a green marker, so pretend that this is green. This is our cucumber. You put it in water that has a very high amount of salt. I don't know the precise salinity of a cucumber, but there's less of it, there's less salt within the tissues of the cucumber's cells than there is in the water surrounding it. So, back to this term. If I can spell it. Selective permeability. It means only certain molecules are allowed to cross the membrane. So let's go ahead and draw a membrane here. This is our hypothetical membrane. And on both sides of the membrane we have salt. These are salt molecules. The salt molecule cannot cross a membrane. It's too big. It cannot pass. But water molecules they're small enough that they absolutely can pass through. So each little black dot I've drawn is a water molecule. How it works is that water diffuses. And we talked about what diffusion was. Diffusion was going from high to low concentration across a membrane. And if you look at the picture that I've drawn, there are way more water molecules here on the right than there are on the left. Which means water is going to move across the membrane towards the side with fewer water molecules. That can be a little tricky to wrap your head around, and so here is an easier way, I think, to understand osmosis. Fewer water molecules means more salt. If there's more salt, you've got fewer water molecules. Which means, in osmosis, water is going to be attracted to or move towards the side of the membrane that has more salt. So water moves from lower salt to higher salt across a membrane. Water is attracted to the salt. How does that apply to a cucumber? You put the cucumber in water, salt water. There's a lot more salt in the water around the cucumber than within its own cells, which means that the water inside the cucumber is attracted to the higher amount of salt surrounding it. It loses water, it shrivels, and in addition to some other chemicals that are involved, you eventually get a pickle. But it has to lose water to get there. And it loses water because you put it in a salty solution that draws the water out towards that saltiness that's surrounding it. this picture shows the same general idea that I talked about. So osmosis causes the movement of water from an area of lower salt towards an area of higher salt. Because again, the salt itself cannot cross the membrane. This describes the whole circumstance in a little bit different way than what I talked about. I'm also going to link a video on Canvas to Bozeman Science video that I think explains it well. So you'll have another video if you're not fully being able to grasp this yet. It can be a tricky concept. But if nothing else, I want you to know that water moves across membranes towards the side with a higher amount of salt. Let's see here. Okay. Tonicity has to do with the amount of salt relative to what is within an organism's body. So I'm going to use fish as an example for this. So let's start with a freshwater fish. Let's draw a fish up here. There we go. So, fresh water, the water you have in Missouri streams, there's no salt, virtually. Salt is measured in parts per thousand. And so, parts per thousand in our streams is negligible, it's effectively zero. Within the tissues of a freshwater fish, I don't know the exact amount of salt, but I know that it's greater than zero. And you know what? To make this work better, I'm going to write these in red. So, no salt surrounding it, greater than zero within. Which means more salt in the fish than outside the fish. Water is attracted to the saltier tissue of the fish. It enters the fish's body. The freshwater fish keeps gaining water because of osmosis, because water is attracted to that salt. Freshwater fish have to expel water constantly in order not to bloat with too much water. The name of this type of solution is a hypotonic solution. It means that the fluid around the cell, or in this case the organism, has less salt than what's actually inside of the organism. Now let's go into the ocean and let's take a look at a starfish. So ocean water, in terms of salinity, amount of salt, is around 35 parts per thousand. Starfish have tissues that match the ocean's salt content. So, water is moving in and out of the starfish, but it's moving equally both directions because we're at equilibrium already. And when you're at equilibrium, you are in an isotonic solution. The fluid surrounding the cell or organism has the same amount of salt as the cell itself. Saltwater fish, on the other hand, saltwater fish, terrible picture of a fish. Saltwater fish, they live in the same environment, but they have less than 35 parts per thousand in their tissues, which means water is attracted to the saltier seawater and leaves their body. So they have to drink seawater frequently, and then they have glands that help excrete the salt. But they have to drink seawater to replenish what they lose because of osmosis. This is also why you don't want to put a freshwater fish in the ocean or vice versa, right? Because if you take a saltwater fish and put him in a lake, he's still adapted to drinking water to make up for the loss. That in freshwater, he'll start drinking the water, plus he's going to gain it now because of osmosis, and then the fish will bloat to death, basically. So that's why fish are very adapted to different salinities, different amounts of salt, because osmosis affects them, and they have to be adapted to it. By the way, the saltwater fish, she lives in a hypertonic environment where you've got more salt around the fish than within it. And these pictures here show this tonicity concept within a single cell as opposed to the fish example. But I think the fish example can be helpful. And that might be the next slide here as well. Yeah, so in a hypotonic solution, that's our freshwater fish, osmosis causes water to enter the cell. Freshwater fish, they just urinate a lot. That's how they get rid of all their water. Some organisms, like this guy here, a little protist, they have chambers that absorb the water that comes in and then they shoot it out. So if you're a freshwater creature, you got to deal with this. You got to deal with water coming in that you don't need. Isotonic, that was our starfish, all right, same amount of salt inside and out. Hypertonic is our marine fish. Again, they lose water due to osmosis, and so they have to drink water in order to compensate, and they get rid of all the excess salt. You and I don't have mechanisms to get rid of excess salt, so if you drink seawater, you actually lose water due to your inability to manage that salt content within your body. This graphic just shows what happens to your individual cells when you expose them to different tonicities. Your blood cells, for example, your red blood cells, within blood vessels, they're surrounded by plasma, which is mostly water, and you gotta have equal amounts of salt inside your blood cell as what's surrounding it, or else your blood cell is going to gain too much water or lose too much water. And plant cells, you can see there, plant cells do best with a hypotonic solution because they want to fill up with water and become rigid, and they've got the cell wall pushing back against that water pressure, which they won't burst like your cells would. Let's see. All right. So all of this osmosis that we've talked about, all of it is occurring without any energy input. Osmosis is passive. And if you see passive, it means no energy input is needed. It just means that as time passes, it's going to happen, right? Because all of this that you're seeing is ultimately water moving toward trying to get to equilibrium. That's what it's all about. And so osmosis occurs passively. However, sometimes you gotta go against the grain. And this analogy that I'm about to show you isn't perfect, but I think it'll help you kind of visualize the general idea. So if you've got a hill, right? If you've got a hill and you want to go down the hill, once you get in that downward trajectory, you can go down the hill without expending any energy. And think of it like biking, right? And I'm not going to try and draw a bike because it would look awful. But if you're going to bike down a hill, you don't need to expend any energy going from a high to low elevation. None at all. It's totally passive. Just no energy needed, you just coast. Going up the hill requires energy. You have to expend a lot of energy to go from a low elevation to a high elevation. Likewise, if you're going from high to low concentrations of molecules, it's passive. It's like going downhill. High to low, no energy expenditure needed. But if you want to go in reverse, low to high, just like going from low to high elevation, this needs energy to do it. And so the opposite of passive, which is no energy needed, is active. So active transport involves molecular movement from low to high concentration. And generally, with active transport, you're going to try and prevent equilibrium from getting reached. You want to keep it uneven in terms of one side of a membrane having more than the other. So, active transport involves moving molecules from low to high against the concentration gradient, and to do that, you got to use energy. If you remember from the previous lecture, we talked about the mitochondria and its job, and the mitochondria's job is to convert food energy into usable energy in the form of adenosine triphosphate, or ATP. ATP is the energy currency of your cells. This is what they can actually use to do stuff. And by do stuff, that includes active transport. So ATP gets spent, much like you spend money, except in this case you spend it on things that require energy. And one example of a process that involves active transport is the sodium-potassium pump. So in order for your nerve cells to properly function, you have to have a certain distribution of sodium and potassium within your cell. And I'll go ahead and draw that up here. I recommend that you watch the video that I've got linked in Canvas for this. But here's the general idea. Here's a cell, okay? And again, K is potassium, Na is sodium. Both of these would have a plus. I don't feel like drawing that every time, but they do have a positive charge. And so within the cell, you've got a lot of sodium ions. I said sodium, didn't I? I meant potassium. You've got a lot of potassium ions, and you've got just a few of them outside. Sodium outside the cell is a lot more abundant. Than it is inside. So if we were to let this be and not put any energy towards it, both sodium and potassium would move to try and reach equilibrium. which means potassium would try and move out, sodium would move in. That would happen without any active transport to prevent it. So what your cells do is they spend energy to keep pumping potassium back in and sodium back out. You have to keep the ratio that you see here for proper nerve cell functioning. And so you got to keep stopping equilibrium from happening. And you can only do that by putting energy in to prevent it from getting there. So I recommend that you watch the video that talks about this in a little more detail. But sodium-potassium pump, good example of active transport within the cell. All right, last slide. So what we've been talking about so far is molecules moving across the membrane, but they've been relatively small. Sometimes molecules are so large that they can't fit through a channel protein. It's really not efficient to try and move them that way. And so instead, you bring them in using a vesicle and trap them inside the vesicle and move them into the cell that way. And so active transport uses transport proteins, but large molecules simply don't work well that way. And so they are brought in by what's called endocytosis. If you break down the words, endo means inside or within, and cyto means cell. So endocytosis involves bringing stuff into the cell. Endocytosis is the formation of a vesicle or a vacuole that transports molecules into the cell. For example, if you eat food, there's a lot of digestive processes that go into breaking it down before it gets small enough to be imported into the cell like this. But once you've got the remnants of your food small enough to be imported into the cell, then your cells will do something called phagocytosis, which involves bringing a solid food particle into the cell by the formation of one of these vesicles or vacuoles, like you see there. Pinocytosis does the same thing, except instead of using the vacuole for a food particle, you're just bringing in fluid. If you ever drink Gatorade, this is kind of that idea. There's important stuff out here surrounding the cell in terms of dissolved substances, ions, the electrolytes, whatnot. You want to bring a bunch of them into the cell. Instead of having them come through and transport proteins, you just make a big vesicle like this that takes in a good chunk of them within the vesicle itself. So pinocytosis involves fluid importation to the cell. Phagocytosis involves bringing in solid particles. Exocytosis is just in reverse. It's when you send something out of the cell, which makes sense. Exo means leaving, exiting, so exocytosis involves reverse of endo. Just send stuff out of the cell. That should finish the cell membrane topic.